Alright guys, so this is our last go through, and then we have a final examination tomorrow. Remember the basic structure, 50 multiple choice questions. It will be on a Scantron form. Just come on in with your number 2 pencil, have a seat, and we'll get started tomorrow, and it'll be just like the midterm in structure and function. The only thing that will be different will be the topics. In terms of computations, there's anywhere from about 5 to 7 on there. Similar to maybe maybe less than 7, like 6 or something. 5 or 6 of them on there. Relatively short number, small number, and they're not that difficult. If you did the work and you put in your time in the homework, basically doing the computations, that should be an easy go for you. Remember though, some of them do take some time. If you're plugging through a Hardy-Weinberg equilibrium, it can take some time, so definitely plan for that. Schedule it in terms of your page pace. There's plenty of time to complete the examination, but if you dawdle, then you can be pressed at the end. Remember to maximize your score. Do the stuff that's easy first, that you know and you know well, and then if you come to something, don't just sit. A lot of people will come to a question they don't know and just sit on it. It doesn't make a lot of sense to do that actually, because what it does is when you sit on a question, it kind of kills your momentum. And then you obsess about the question, and then if you blow it or you're not sure about it, it can alter everything that's coming after that. So you might, give up easy questions that you know well because now your anxiety level is up. So if you come to something that's trickier, you don't know it, move on. Answer the stuff that you know as well as you can while you're nice and calm. And then leave the stuff you don't for the very end. And then don't change answers unless you have a good reason to. I see a lot of people who get the right answer because their gut's telling them it's the right answer, and then for some reason they convince themselves it's not based on any facts or anything, just some weird feeling. And that always usually ends up poorly, okay? So if you have a good reason, oh, I remember now, yeah, go back and change an answer. But if you don't really, leave it where it was. Most of the time you're correct in the first place. So think smart when you're taking an examination. Taking an examination, there's a lot of gamemanship that goes into it. So it's important to have a strategy for how to take an examination. I see lots of people who know what they're doing, but they get test anxiety, right? And that's a shame. So there's lots of ways you can reduce that anxiety. The first thing is to study, obviously. The second thing is to try and pre-test yourself a little bit, reduce the overall stress levels. That always helps. And if you come in, and you're informed and confident, you're less likely to freak out. But when people are freaking out around you, having that skill set to kind of block out the world is really important. So think about how you want to do that. And like I said, going through the examination at pace and avoiding questions that are going to throw you and coming back at them at the end, that's a great way to do it. Because if something's going to throw you, I'd rather have it throw me in the last 5 minutes where I've answered 90% of the examination already and did well. Then the rest of the stuff's just kind of gravy on the end. So think smart when taking an examination. If you have questions about a particular question, raise your hand. I'm happy to try and clarify as much as I can. Nothing's designed to be tricky. But sometimes when you're dealing with example-based exams, they can be a little dense. So it's like, what the hell is he actually asking me? Right? So I'm happy to clarify what I'm actually asking for in a particular question. I'm not going to give you an answer, but I'll definitely clarify a question and tell you what I'm looking for. Does that make sense? Don't waste points just because I don't understand this question. Well, it must be A. Well, that doesn't make a lot of sense. Ask for clarification on the question. You might get some insight that'll help you to deduce what the proper answer is. Right, anything else? Okay, well let's finish it up then. So we were basically talking about the last step in the evolutionary process, which is speciation. So let's kind of run through our basic mantra again. So we start off with what? The microevolutionary process? Variation acted upon by natural selection leads to genetic conversion supported by reproductive isolation, which can, doesn't have to, but can lead to speciation. All right. So we started talking about speciation. Now speciation might intuitively look like it's the simplest thing in the microevolutionary process and the most straightforward, but it's exactly the opposite of that. It's really difficult to decide where one species ends and another one begins. So that's a really tough thing. So we can look at a phylogeny. What's a phylogeny? An evolutionary what? History. It's an evolutionary history of organisms, okay? And that can give us some insight about particular groups. When looking at phylogenies, if I was to look at a bubble plot of a phylogeny, if I look at the bottom of the graphic, what is that? The past or the present? That's the past. The top is present. The width of the bubble indicates the diversity. The length of the bubble indicates how long it has existed. Bubbles that pinch out cleanly and sharp points mean what? Natural extinction events. Ones that get lopped off mean what? Some kind of mass extinction event. All right? So I started talking about different concepts of how to explain when you have a species. For sexually reproducing organisms, it's fairly easy. What is it? When you can't do what? You're a new species. When you can't reproduce with the old species, you can't donate genes anymore back and forth. If that's the case, well, you're considered to be a different species, right? What's that species concept called? The biological species concept. So it was developed in the 40s. It still works. It works great. What's the problem with it? It doesn't account for, 2/3 of what's alive on the planet. It only accounts for organisms that are sexually reproducing. So asexual reproducing organisms doesn't work well at all. So we have other concepts that we can use. What's the one where we look at the morphology? Well, I just gave it away, didn't I? You look at how something looks in terms of its traits, that's what? The morphological species concept. Does it work? Very iffy, okay? Because why does that one often fail? Because there's tons of variation within individual populations. You can see they're same species, but depending on whatever environmental gradient they are, they look completely different. Right? You'll have something that'll be this big, something that's almost a big giant tree. They look completely different in terms of plant. Exactly the same thing. It's just the water or the soil that's different. So that one's kind of tricky and it has lots of problems. What's the one where what do we do for dead things that are no longer here? All we have is some remnants of what was once bones. That's the paleontological species concept. Obviously, what's the problem with that? Not everything fossilizes. That's exactly it. Not everything fossilizes. And since we only have partial fossil record, what's that? Sorry. It's very limited evidence, exactly. And then also, here's the thing. What if it died when it was a baby, and then it died when it was an adult? We might think they're completely what? Different species. That's exactly right. So the fossils are very, very tricky. What if I was interested in looking at the niches and how they overlap and don't overlap? That would be the ecological species concept. What's the problem with that one? How the hell do you define an ecological niche? And also it assumes that the niches are relatively static in terms of their nature. So remember that I described a niche as kind of an n-dimensional hypervolume. That was a fun, a big wordy thing that basically says you have all these axes of resource utilization. They create a spherical kind of space around you ecologically. But is that a static space? No, it's constantly expanding and contracting and moves around. It's a lot like you guys, when you were kids, you ever blow those big giant bubbles? And they go like this with them and they change their shape. That's what a niche actually does. It's not this perfectly round sphere like that. It's moving, contracting, and it's quite fluid. Well, if I have to decide if you're a species and all of a sudden your niche is way over here and the next minute it's back over here, that makes it tough to decide whether you're a species or not based on the ecological niche space and where it overlaps. So that one's tricky as well. I have a nice set of brand new technological ways to identify differences in species. And what are those referred to collectively as? What is it? The phylogenetic approaches. I gave you two of those. What are they? The most common ones? Not the phylogeny, but the two techniques. Molecular clock, which takes a look at the number of what? Neutral mutation accumulations. And what else, the other one? DNA hybridization, right, which basically looks at how much energy is required to split the bonds between hybridized DNA. They're great. They really work well. What's the problem with those? They'll give me an exact percentile difference between the two species. I should say two species, between the two groups I'm interested to know if they're species. But what's the problem? What's that? I don't know what the cutoff should be. Is it 10%? Is it 1%? Is it less than 1%? So that's really problematic. I just don't know where to set it. So we have to figure that out. So often what do? you use all three, you use all of them and see which ones are corresponding to each other. And if you have considerable overlap among the groups, yeah, it's probably a species, right? Probably a different species. If maybe only one of them showing it, maybe I'm a little more hesitant to make that call. All right, so take home is it's a tough thing to decide if you have a species or not. So I think we started talking about the speed at which you can create a species, and this I should say form a species. And so in this particular case, it can be fast or it can be relatively slow. And I think we stopped at allopatric speciation. Is that where we stopped? Yeah. Okay. Let's pick that up. Adaptive radiation. So I went past allopatric speciation. What is allopatric speciation then? Physical barrier separates the populations, promoting genetic divergence, reproductive isolation, and ultimately promotes speciation. There you go. Okay. And so I gave you this example in terms of small isolated populations. Small isolated populations are much more likely to undergo allopatric speciation because they're primed and set for it, because what's going to have a big effect on them? Genetic drift, mutation, natural selection, all those things will operate at an enhanced degree when the population is small. And I gave you a nice example of this with an adaptive radiation. And adaptive radiation means what? You get some precursor species that gets there. When it gets to the new habitat, what does it do? It radiates out quickly to take advantage of the new habitat types and resources that are available, quickly resulting in divergence in morphology, physiology, or behavior. That's adaptive radiation. All right, that brings us up to speed. Yes?
Yeah.
So let's talk about the pace of allopatric speciation and its timing. So when we think about geographic distances, they can often separate species. And so gene flow, again, is really critical. Gene flow does what to the differences between different populations in terms of alleles? It homogenizes them, it brings them back together. So when we actually look around in the world, we see that often gene flow is not like this gushing river, and it's often not completely zero. It's just this intermittent trickle, like a drip out of a faucet, right? And so often there is some connectivity, but it can be relatively limited in nature. So we can get rapid speciation by some kind of punctuated equilibrium event that radically changes the environment. So barriers arise all the time, and they can occur quite rapidly. Sometimes we think of them as like slow, like the rise of a mountain range, like the Rockies or something like that. But often it can be overnight. So like an example, in the 1800s there was a massive earthquake in the Mississippi Valley. And so basically, actually along the Mississippi line. And what it did was it actually was so large, it shifted the course of the Mississippi. We don't see many earthquakes like this across the central part of America. It's funny, if you're a Californian, it's like, if you get an earthquake, you're kind of like, oh, there's an earthquake. Have you ever seen somebody from like the Midwest when they're here and there's an earthquake? It's like full freak out, right? They're like on the ground, holding on, screaming and yelling. You're like, what is your freaking problem, right? It's just because you're accustomed to it. So, but occasionally you do see earthquakes there, even though they're much rarer than they're out here. We get all kinds of earthquakes because why? Yeah, we're sitting right on a massive fault between two gigantic plates, the Pacific plate and the North American plate, and they're sliding past each other, right? You guys know what an earthquake is, basically how it's formed? So when plates come together, did we ever talk about this? When plates come together, two crustal plates are lighter than oceanic plates, right? So crustal plates come together, what do? They lift up. They create fold mountains like the Himalayas. Oceanic plates are super heavy and dense, so when they hit, what do? They subduct and they go down. And typically when you have oceanic against a crustal plate, what will happen? The crust will lift up and the ocean one goes underneath, or they can butt up and slide past each other. And earthquakes are basically the release of energy. So if you put your hands together like this and just go easy, it's super easy to move. There's some friction, right? But now what you do is put more pressure on them. It's a lot harder, right? And when they do release, you'll get a lot of energy that slips and slides. That's basically how an earthquake is. So you got the pressure of the plates pushing, pushing, pushing. Then they slip. And when they slip, it releases all that energy. And depending on how shallow or deep it is in the crust, that's where the severity of the earthquake will result to us. So in this case, it actually was so strong, it shifted the Mississippi River. And when it did that, it cut off populations that were once contiguous. And what happened was they quickly diverged. So basically, You had insect populations on both sides, terrestrial insects but that weren't winged at this particular part of their wings were not that functional. And so what happened was, they were sitting on each side, river shifts, now they're isolated in a different way, and then when the river changed back to its original course, lo and behold, enough divergence had occurred that reproductive isolation had kicked in and they were no longer able to breed with each other, therefore they are what? New species, that's exactly right. So ice ages and interglacial periods, rise in mountain range, those are more gradual types of speciation where the barrier doesn't come up really instantaneously like the flow of a river moving around, but they come up slowly over time. And then what they do is they start to slowly impede the migration between the two populations. We can take a look at this in the fossil record, we see it all the time. A nice example of this are the birds we talked about earlier. Remember the rhea and the emu and the ostrich? They're similar yet different. Well, why? Because at one point they were all together. But what's happened? They've rafted apart via continental drift, and now they're adapting to their local environments, and we're seeing divergence. They don't breed with each other anymore, they're different species. All right, so sympatric speciation is exactly the opposite. It's not separation, but it's together. So a species may form within the home range of an existing species in the absence of any physical barrier. And so there are a couple different ways you can do it. You can deal with genetic mistakes like polyploidy. You can adapt to local microclimates or microhabitats within a larger habitat whole. And then sexual selection is another way that you can drive it quite easily. So we want to spend a little time talking about polyploidy. I don't really want to get into much the kind of the genetics of it or anything. I just want you to have a general understanding of what happens. Did you guys talk about this at all in 1A? Mistakes in chromosomal separation at the meiotic divisions. Did you guys talk about this a little bit? I don't want to go into it too deep. I just want you to have a general understanding of how it affects the evolutionary process. So autopolyploidy is basically a non-disjunction of chromosomes during gametic formation in the same species. Remember that homologous chromosomes line up, right? They can exchange information by crossing over. And then what they'll do is they'll separate and they go into individual gametes, right? Well, what happens is you can get mistakes because they're so similar molecularly, they can stick together. And so what happens sometimes is they don't have a disjunction and separate, they stay together, and then the joined chromosome goes into one complete gamete itself. That's a big screw up. Because, for instance, for you to have a human intact child, right, what do you need to have? How many chromosomes? 46 is the human count, 23 from mom, 23 from dad. But what happens is if those don't basically disconnect, it ruins the number, and that causes all kinds of problems for us, right, as a species. You have all kinds of, in certain cases, maybe you have one extra, and that creates a whole series of problems. But any more than that, you have really severe problems, usually lethal, okay? Plants can handle it a lot better than animals can. So basically, an individual that has more than 2 chromosome sets, all derived from a single species, can result in speciation. So let's talk about how this actually works. So this would be an example of autopolyploidy. This is intraspecies breeding, all right? It produces a 2N diploid individual to a 4N individual because they don't detach. So it's supposed to go 2N, and you're going to create 1N back to 2N, right? But in this case, they don't detach, so your individual at the end ends up being 4N. It's double the number. So in this particular case, can you ever get a successful reproductive event between the diploid individual and the tetraploid individual? You can't. They don't line up. The number count isn't right. So they're completely sterile in that case. We get total reproductive isolation between the original organism and then the tetraploid organism. It doesn't work. However, for animals, you're done. Okay? But for plants, lots of plants can self-fertilize. All right? So a 4N plant, you get this tetraploid, and it can actually produce fertile offspring via self-pollination, or it can mate with another 4N individual itself. So what do you get in that case? You get a brand new species, okay? It's self-replicating itself. So in one generation, autopolyploid can generate a speciation event. You went from a 2N individual to a 4N individual. The 4N individual can't replicate or can't reproduce with the 2N individual anymore, but it can self-pollinate, creating itself, right? Or it can go 4N, 4N and be fine too, as long as there's multiple individuals that have that genetic component. So that's one way you can create a brand new species. And here's kind of a nice schematic of it, right? You start off with 2N, there's 6 individual chromosomes. They go unreduced, but there's a meiotic error where they stick together. That creates the foreign, which is 12 of them. Then those ones there are brand new species, and then they can basically self-fertilize or find another foreign individual to complete their reproductive process. There's another type of polyploid, and this is allopolyploidy. What does that mean, allo? Auto means what? Self. Allo means different, okay? So in this particular case, this is also quite common. This is an interspecies breeding, not an intraspecies breeding. So inter means what? Two different species. Intra meant same, the same, within the same species. All right, so interspecies breeding produces a hybrid. What do we know about hybrids? They're usually sterile or infertile, okay? They're usually sterile or infertile. So in the hybrids, you have a set of chromosomes from each species, but again, they're sterile or infertile with each other, and they can't interbreed with the old original parental species. However, this is where the meiotic error then kicks in. So the sterile hybrid can propagate asexually. So many plants can do this. They can propagate asexual, generating a speciation event. Or in subsequent generations, the sterile hybrid can change into a fertile polyploid via exactly the same mechanism that we saw in autopolyploidy, where you have that disjunction of the chromosomes. You get the meiotic error, and lo and behold, they can self-fertilize, or they can find another individual with the same genetic component, and then you can have a new species. So it's basically like skipping a step. So what are the differences here? This one occurs, auto occurs within one species. Allo occurs between two different species. In this case, you get a meiotic error, which results in a doubling of the chromosomal number. That individual can then self-fertilize or basically have a reproductive event with another individual of the same chromosomal number. Here, you create a hybrid from the initial cross, which is there infertile, right, or completely sterile. And then you get a mitotic error in that, and now it can self-fertilize, or it can find another individual of the same genetic number, and then you have a new species. So it basically is skipping, it's going one extra step. Make sense? So same species, different species, one extra piece, boom, there you go. And so that's the difference between auto and allo. And so they can result in new species within 1 or 2 generations, right? So in this case, 1 generation, boom, you have it. In that case, it's 2 generations. So that's how it can work, and you can get an immediate speciation event. That can happen in plants, and it happens quite commonly. Just to give you an idea, many of the things that we eat as domesticated agricultural crops are allopolyploids. And they've been bred over and over again. Oats, cotton, potatoes, tobacco, wheat especially. Wheat is a kind of a genetic nightmare of different kinds of breeding. So they're all polyploids. Wheat itself is basically an allohexaploid. It has 6 sets of chromosomes. The first cross was thought to happen naturally in the environment, and then we've repeatedly crossed it to create, basically if you see the precursor to wheat, wheat is a what? It's a grain, which means it's just a grass, okay? So you can find kind of the precursor of domesticated wheat. And what do we harvest from wheat? So basically, if you see grass, it wants to give seed, right? And it has that little tuft on it that prevents the seed. Well, what we've done is we've artificially selected for the size of that. So when you see wheat, it's got this big seeding pod right on top of it, and that's what we harvest. And so basically, just manipulation, artificial selection for that has led to the product that we have, which is wheat today. We've modified wheat so extensively that a lot of people have problems digesting it anymore, right? Because it's been modified so intensively that some people have real problems digestively with it. Polyploid in animals is a rare event. You see it in some frogs and some fish. You rarely see it anything above that. There's other mechanisms of sympatric speciation as well. And so basically, you have some kind of genetic factors that enable a subpopulation within a greater population to exploit a particular resource in the environment, not used by the original parental group. And so over time, those genetic differences are going to build, they accumulate, and that's going to lead to reproductive isolation and speciation. So here's a nice example of this in terms of sympatric speciation, and it's parapatric speciation. So in parapatric speciation, you simply have adaptation within a local environment, and typically sexual selection promotes the speciation event. what you often see is it creates neighboring populations that become distinct species while they maintain kind of a tight boundary of contact along the common border. So they're in contact with each other, but yet speciation is occurring. Right on that borderline, you often get hybridization between the two groups. So when you take a look at this, a nice example is actually in cichlid fishes in the Great African Rift Lakes. And so it's a really nice example of parapatric speciation. So what happens here is you can get little tiny subtle differences in the fishes based on morphological characteristics. So let's say you have a big group of one particular species of fish, and a mutation arises that gives one a red spot. Well, it might just be that certain fish within the group like the red spot, and they mate with that fish selectively. So what's going to happen to that red spot? It's going to pop up more in the next generation. And then what you'll see is red spot fish will only mate with red spot fish. Now, when I look at it superficially, is there really any distinction in the habitat? There's none. It's just a big lake. It's all a contiguous water body, and they're all right together. But it's the presence of that one little spot that starts to divide the population, and before you know it, I got all the red fish right here, and I got all the non-red fish right here. There might be a couple hybrids in the middle, but I have a new species. They're not breeding with each other at all anymore. So something as simple as that, a little dot of something on a fin, can radically shift the subpopulation into speciation events. And that would be a nice example of parapatric speciation, which is speciation in the contiguous environment sharing a border. All right, so let's kind of sum it all up. We've run through the microevolutionary process in gory detail. We've run from the start to the finish. Again, it's not the whole story, far from it, because we've excluded much of the living world, right? And you're going to pick that up as you go into upper division based on the courses that you are taught. If you go into microbiology, you're going to learn a tremendous amount about the evolution of bacterial life forms, fungal life forms, because you'll have to figure out how to defeat them. It's not an easy thing, and their evolution rates are rapid. And you'll learn the ways in which they transfer genetic information and enhance their evolutionary speed of change. But they change in all the same ways, morphologically, physiologically, and behaviorally. So we're interested then is, well, what happens now? I've generated a new species through this process. Okay. So basically, speciation is really one endpoint, but it's also a starting point. It ends the microevolutionary process, but it's actually the start of the macroevolutionary process. So microevolution started with these little tiny differences, right? Little tiny things, small genotypic and phenotypic differences were selected for, which resulted in large changes in the overall population. However, once you get those changes and are significant enough and you form a new species, you're going to see that those species diverge again, and again, and again, and again. And those differences become more pronounced over time. So what started out as the difference between a red dot and no dot, all of a sudden became the difference between a fish that's 20 pounds and a fish that's 10 pounds, and a fish that's on the bottom and a fish that's on the top, and a fish that has a different color fin and a different shape fin. And before you know it, you have fish that are walking around on the bottom, and now they can walk on the surface to get from pond to pond. And these differences start to become greater and greater and greater. How much time has there been for this stuff to occur? How long? 3.8 billion years. Is that plenty of time for stuff to change? It got radically different? Yeah, you better believe it, okay? So again, this is going to happen over and over again and become more pronounced through time. So speciation then constitutes the beginning of macroevolutionary change. These are the large-scale patterns that you see in the fossil record. They're trends and rates of change among family levels and much more inclusive groups. So it's kind of the idea of, well, how the hell do you get an amphibian? Where did that come from, right? How do you start off with a single-cell precursor, choanoflagellate, that's the precursor of all animal life, and all of a sudden I'm looking at amphibians and mammals and birds and all these kinds of crazy organisms, widely divergent, but yet sharing many characteristics at the same time. And then I have other kingdoms. I got plants, and I've got microbes. I got eukarya. I have basically archaea. On and on and on, right? So how do you get these major differences in the groups? That's the story of macroevolution, and that's EEMB3. So now you know basically the starting point, which is speciation, and EEMB3 will walk you through the macroevolutionary parts, which are the formation of all those different groups on the planet, from microbial life to plant life and through animal life. All right, so when we started, we were confronted with this diagram, right? Which again is a cool diagram, but what's the problem with it? It's inaccurate not in what it's showing you, but in how it's being perceived, correct? And we don't typically make that correction, because again, what is it insinuating? That evolution is a linear sequence of A to B to C to D, E, and it's not. It's what? It's a branching crazy tree. If I was to take one of those sequences, it'd appear kind of linear, but remember, we're always going back to common ancestry. So the things that are around us today, we always had common ancestors with something in the past. It's not like I evolved from chimpanzee. It doesn't work that way. And that's basically what that's insinuating. So again, what was your homework?
Teach someone.
To educate people that it's not that, but it's this, and it's this, and those are just snapshots along the way. You go back 30,000 years ago, plus, were we the only humans on the planet? We were not. Who else was here? Neanderthal. You go back a couple more 100,000 or 100,000 years more or so, now who's here? You got us, you got Neanderthals running around. No, I should say 100,000, but a little further back than that. And you're going to get different species. You have 3 species of humans on the planet. We live in a boring planet, human-wise. What happened to the rest of them? We probably killed them. That's kind of what happened. But they're not here today, right? So these are all snapshots along that kind of route. So in summary, remember, all species past and present are related by way of common descent. 3.8 billion years of that descent. Life is the story of species. If you still have traits that started back 3.8 billion years ago, why do you have them? They still work, and if it ain't broke, don't fix it. And if you have different traits that are new and innovative, it's because you've adapted to a new environment on the planet that wasn't here before. So that's the basic idea. Traits persist, they've been lost, or they've been modified over time. And then life is also the story of the macroevolutionary process and large-scale changes in the groups which will experience in the EEMB3. So before you leave, remember, you hear a lot of crazy stuff out there. Most of it's inaccurate and wrong. Today I had to listen to someone rant all the way in on the radio about the hoax of climate change on a nationally syndicated radio station going out to millions of people. Completely inaccurate, basically a lie. I'm not even saying inaccurate, just lying. We're in a weird time where if you say a lie, and you say it loud, and you say it enough times, it becomes somebody's truth. We got to stop that. So it's your job as scientists to not only speak amongst yourselves, but to educate the general population. And this is a great place to start. All right? So best of luck on the examinations. That's what I'm